Hello students, how are you all? This video is in continuation with Purine biosynthesis part 1. I'm sorry about that. There was technical glitch in that video. So I already discussed with you that there are 10 steps in the purine biosynthesis starting from PRPP to IMP. So the step in which PRPP synthesized is not included in the de novo synthesis, as this PRPP is basically the donor of ribose 5-phosphate moiety, and this is also PRPP is also utilized for the synthesis of nucleotide coenzymes and as well as in the salvage pathway also. So the very first step, so there are two rings in the purine. So first we will synthesize this ring, second ring. So first of all, phosphoribosylamine is formed in this glutamine, this N9, that is the last nitrogen atom that is added first. So glutamine, in this very step in which phosphoribosylamine is formed, this is the rate limiting step, we can say, in which enzyme is glutamine PRPP amidotransferase. So glutamine donates its amide nitrogen to PRPP, displacing its pyrophosphate moiety in presence of an enzyme glutamine PRPP amidotransferase. So this is the rate limiting enzyme or the regulatory enzyme of this pathway. Then from and secondly, I told you that wherever we are using synthetase enzyme and the ring closure steps, we need ATP. So ATP is required in those steps. So from PRA to GAR, PRA is phosphoribosylamine. From PRA, that is phosphoribosylamine, to GAR, GAR is glycinamide ribonucleotide. In this step, glycine is added. So addition of glycine is taking place in this step. So C4, C5 and N7, they are contributed by glycine in presence of an enzyme GAR synthetase. So again ATP is required in this step. Then from GAR to FGAR, this formylation is taking place in this step, and the enzyme is formyl, this GAR transformylase is the enzyme in this. So this N10 formyl tetrahydrofolate donates its formyl group to GAR to form FGR, that is formyl glycinamide ribonucleotide. Then from FGAR to FGAM, another glutamine is donating its, this is the second glutamine, amide nitrogen of glutamine. This glutamine transfers its second amide nitrogen to FGAR to form FGAM. Then FGAM is formyl glycinamidine. After the this thing, FGR to FGAM, FGAM is formyl glycinamidine. Then from FGAM to this AIR, this is a ring imidazole ring closure step. This second ring is closed in this in presence of an enzyme AIR synthetase. So again ATP dependent reaction, this AIR is formed. Then in the next, from AIR to CIR, carboxylation is taking place in this. So in presence of an enzyme AIR carboxylase, so second ring is now built by stepwise addition of the final three atoms. Now the remaining atoms are this C and 1, then this 2, C2 of format. So now the first atom to be added is the C6 carbon now, which comes from CO2, which is incorporated to form CIR. So this carboxylation is something, exception is there in this carboxylation reaction, because carboxylation are of two types, Barton dependent and Barton independent. So over here this carboxylation is Barton independent, so we do not need ATP or biotin in this carboxylation reaction. then from CAIR to SAICAR. So in this aspartate addition is taking place. This amino group of aspartate is added in this in presence of an enzyme SAICAR, that is succinyloaminoimidazole carboxamide ribonucleotide synthetase enzyme. So this N1, we can say N1 of purine is contributed by the condensation of aspartate to CAIR to form SAICAR, but we only want the amino group of aspartate. So after that, subsequently the fumarate is cleaved in this reaction, and in presence of an enzyme adenylosuccinate lyase, and we get this AICR, amino imidazole carboxamide ribonucleotide. So after this, again this formylation is taking place in this reaction with the help of an enzyme formyl transferase. So the last purine ring, we can say purine carbon, is added as a formyl group by formyl transferase to form FAICAR. So in the final step, again the ring closure. So IMP cyclohydrolase catalyzes this closure of the second purine ring to form IMP. Now from IMP to AMP and GMP, as we do not want this IMP, we want AMP and GMP. So for conversion from IMP to AMP, the C2 of IMP, this we can say this amino group of this aspartate, is first linked to this IMP to yield adenylosuccinate. This is an intermediate which is formed, and this trap is driven by this GTP hydrolysis in presence of an enzyme adenylosuccinate synthetase. So in the second step, this fumarate is cleaved, as we do not need whole of this aspartate. We only want the amino group of aspartate. In the presence of an enzyme adenylosuccinate lyase, AMP is formed. uh, in the presence of an enzyme, this adenylosuccinate lyase, so in this, from IMP to GMP conversion, C2 of IMP is oxidized by the IMP dehydrogenase, which is an NAD dependent enzyme, to form XMP as an intermediate. XMP is then converted to GMP by using glutamine as the nitrogen donor, and the reaction is powered by ATP hydrolysis. So now we have AMP GMP, okay, with us. So this from AMP to ADP, we need kinases, and from GDP to GTP, we need again kinases. So there are nucleoside monophosphate kinases are required. Monophosphate kinases are required, in which this AMP is converted to ADP and GMP to GDP. This diphosphate kinases convert GDP to GTP or CDP to CTP. So now we have AMP, ADP, ATP with us, and GMP, GDP, and GTP, this GTP with us. So all these are the ribonucleotides. So these ribonucleotides will synthesize RNA, but we want DNA also. So for the conversion of this ribonucleotides to deoxyribonucleotides, we want another enzyme. We need another enzyme that is known as ribonucleotide reductase. Okay, so in this ribonucleotide reductase enzyme is this is an enzyme that converts the ribonucleotides to the deoxyribonucleotides. Okay, so this enzyme requires a peptide coenzyme thioredoxin, which is the donor of the reducing equivalents. So during the process, we can say this thioredoxin reduced is converted to the oxidized form. So subsequently, it is reconverted to the reduced form by the reducing equivalents of NADPH. So thus, in this series of reactions, we can say this NADPH is the ultimate donor of the reducing equivalent. So this enzyme, you should know that this converts all the ribonucleotides to the deoxyribonucleotides. This is specific for a variety of nucleoside diphosphates, that is purines ADP, GDP as well as pyrimidine CDP, UDP, converting them to the corresponding deoxyribonucleoside diphosphates, that is DADP, DGDP, DCDP and the DUDP. Then next is the regulation of purine biosynthesis. So the de novo synthesis of purine nucleotides is finally coordinated to maintain the total pool of the purine nucleotides available for the nucleic acid synthesis and to maintain the proportionate concentrations of ATP and GTP according to cellular needs. So purine nucleotides are interconverted to balance the levels of adenine and guanine nucleotides. Then these are the regulatory points. One is the first is the availability of PRPP. So we'll be discussing all these points in detail. All these are the regulatory points. As I told you, this glutamine PRPP amidotransferase, this is the first enzyme, rate limiting enzyme. So AMP, GMP, these are the end products of the pathway. So these end products feedback regulate this particular enzyme, and AMP GMP feedback regulate their formation from IMP, and ATP and GTP formation has a reciprocal relationships. So these are the regulatory points. We will be discussing all this in detail. Number one is the availability of PRPP. As you all know that PRPP is required for the synthesis of IMP, because if we do not have this moiety, we cannot synthesize this IMP. So there is a need. There is a need for this availability of this PRPP is important. So biosynthesis of IMP is regulated at the first step of the pathway by the availability of ribose 5-phosphate and hence PRPP. Now in this, this is the preparatory step in which PRPP is formed. So this PRPP is an activated pentose, we can say, that participates in the synthesis and the salvage of purines and pyrimidines. So synthesis of PRPP is dependent on this enzyme PRPP synthetase, then ATP and also the ribose 5-phosphate moiety. So this excellent enzyme, that is PRPP synthetase, this is activated by inorganic phosphate and inhibited by the purine ribonucleotides, that is IMP, GMP and AMP, the end products of the de novo purine synthesis. So the reaction catalyzed by this amidotransferase, this is the committed step, that is the site of metabolic regulation. So this enzyme is regulated allosterically by the end products of the pathway, that is IMP, AMP and GMP, which serve as all these IMP, GMP, MP, all these serve as negative effectors for this enzyme, whereas this PRPP is the positive effector for the enzyme. Then biosynthesis of AMP and GMP, these from IMP is also regulated. So both AMP and GMP each are competitive inhibitors of IMP in their own synthesis. Then this enzyme adenylosuccinate synthetase, this is the rate limiting enzyme in the conversion of IMP to AMP, where AMP act as a competitive inhibitor, whereas in this from IMP to GMP synthesis, this IMP dehydrogenase is the rate limiting enzyme. So this is regulated by GMP, which act as a competitive inhibitor. Further, the rate of GMP synthesis increases with the concentration of ATP, while that of AMP increases with the concentration of GTP. So this is cross-regulated. As I told you that from IMP to GMP conversion, ATP is required, and from IMP to AMP conversion, GTP is required. So synthesis of adenine and guanine nucleotides are cross-regulated so that their production is always balanced. So conversion of IMP to AMP requires GTP. This is reverse. Conversion of IMP to GMP requires ATP. So this ensures the balanced production of AMP and GMP. Now coming to the next pathway, that is the salvage pathway, salvage synthesis of purine nucleotides. Now in this you need to remember only the two enzymes APRTase, that is adenine phosphoribosyl transferase, as well as the HGPRTase, that is hypoxanthine guanine phosphoribosyl transferase. So PRPP is the starting material. In this way, it is also a substrate for de novo synthesis, as I already told you, as these two pathways are closely interrelated. Which pathways? De novo as well as the salvage pathway, as PRPP is required in both the pathways. So both these pathways are closely interrelated. So the free purines are salvaged by two different enzymes. Number one is APRTase and second is HGPRTase. So the pathway is of special importance in tissues like RBCs and the brain where de novo pathway is not operating. So this has a special significance in tissues like RBCs and brain where de novo pathway is not operating. The salvage pathway economizes the energy expenditure. As you have seen in the de novo pathway, there are number of steps in which we are using the synthetase enzyme and the ring closure step. So we need ATP everywhere. So in this way, this salvage pathway, this economizes the energy expenditure. Absence of enzymes of the salvage pathway produces specific clinical syndromes. This adenine plus PRPP will form in presence of APRTase will form AMP. Then hypoxanthine plus PRPP, HGPRTase will give you IMP, guanine and PRPP. In presence of HGPRTase will form GMP. So these are only the steps of the salvage pathway. So you need to remember both these enzymes, APRTase as well as the HGPRTase. Now coming to the degradation of purine nucleotides, you know that this nucleotidase hydrolyzes the nucleotide to nucleoside as well as the base. Then nucleosides are hydrolyzed by nucleosidases to form ribose 5-phosphate as well as the purine base, and purines are finally degraded to the uric acid. So this is the product of the purine catabolism. Now in this, this is the degradation pathway of purine. So AMP, that is a nucleotide, when it is converted to this nucleoside in presence of this enzyme nucleotidase. Nucleotidase hydrolyzes the nucleotide to nucleoside. Then this nucleoside in presence of this in this deamination is taking place in presence of an enzyme adenosine deaminase. Inosine is formed. Then inosine to hypoxanthine, another enzyme is required, purine nucleoside phosphorylase. So this, as I told you, hypoxanthine to xanthine is converted by xanthine oxidase. So this is the very important enzyme of this pathway, which converts hypoxanthine to xanthine and xanthine to uric acid. In the same way, GMP, that is nucleotidase hydrolyzes the nucleotide to guanosine, that is nucleoside, and this purine nucleoside phosphorylase is converted into the purine base. Then guanine to xanthine by deaminase. Deamination is taking place, xanthine is formed, and in presence of an enzyme xanthine oxidase, xanthine is converted to the uric acid. So uric acid is the final end product of this pathway. So xanthine oxidase is a very important enzyme of purine degradation, which is exclusively found in the liver and the small intestine. It converts hypoxanthine to xanthine and xanthine to uric acid. Xanthine oxidase contains electron transfer molecules like FAD, molybdenum and iron sulfur clusters as its prosthetic group. So molecular oxygen is the final electron acceptor in the reaction and is reduced to H2O2. So this H2O2 is potentially dangerous to the tissues and hence is decomposed to water and oxygen by the enzyme catalase. So this purine nucleotides, that is AMP and GMP, leads to the formation of uric acid in humans, which is excreted in the urine. So as you know, uric acid is not very soluble in an aqueous medium. So elevated level of uric acid in the serum results in the deposition of urate crystals, primarily in joints, a clinical condition that is known as gout. So in the last video of this purine and pyrimidine metabolism, I'll be discussing the disorders associated with purine and the pyrimidine metabolism. So do watch next video on the diseases associated with the purines and the metabolism. In this, I'll be discussing in detail about gout and the Lesch Nyhan syndrome, as well as the orotic aciduria. Thank you.